Um, Good afternoon to you all. My name is Tindyebwa, and I'm presenting the research that I've been doing for the past few weeks. And the title is Development of a Preliminary Secondary Model for Spoilage of Atlantic Salmon. And my supervisors, Professor Mark Tamplin and Dr. Shen, to whom I'm very grateful because they were really very helpful in my research. So before I go into the details, I'd like to mention that this is a preliminary research. It's not the end point, and a little more has to be done. Okay, I'll start with the background. My research was with Atlantic salmon, and which falls into the fishing industry. It's worth billions in Australia, and salmon being the second most important seafood product by production as well as monetary value in Australia. And with that, Tasmania produces about 95% of the total production in Australia. So for that, I thought it was actually quite impressive, and this gives a reason why research on Atlantic salmon is really important. So the research was, of course, it was development of a secondary model to determine fish spoilage. So our main focus was on spoilage. And spoilage in fish occurs at two stages. The first one is immediately post-mortem. That is when the fish has just been caught and it's dead. So the very, very first process of spoilage is by natural biochemical processes. And then the next step is microbial spoilage, which of course goes together with sensory spoilage, because the microbes will impart bad odors and flavors to the fish, and of course reduce its quality. So even the natural biochemical processes will affect fish quality. So that is one step of spoilage, and then the second step of spoilage, which we are also, which we are interested in. So at the beginning of the study, when you have the samples together, when you carry the samples, for example, to the lab, you have microbes already present on the fish. And the microbes present will depend on the fish environment, the aquatic environment from which you obtain the fish. And of course, there are other parts of the fish, for example, gut contents, gills and skin. All those, through processing and handling, of course, depending on the procedures in processing and handling, they will transfer those microbes to the fish flesh itself. So the type of fish is also important. Different fish tend to have different microbial communities, and also the geographical location. So if you have, for example, a subtropical location, the microbial communities will be quite different from a temperate location. So that is how geographical location is important to microbial communities on the fish. So, of course, you have the initial populations. So, at the end of storage, for example, when the fish has gone bad, you have the communities have changed. So, at the end of storage, you have specific spoilage organisms, which will depend. Specific spoilage organisms are the ones that culprits in the actual spoilage of the fish. Others may be present but don't cause any bad effects on the fish or change its quality, but they are present. But then the specific spoilage organisms will be the ones that will actually cause the spoilage that you can detect through their growth. Of course, they'll occur in high numbers, and then also they impart maybe bad odors to the product. So to determine the specific spoilage organism, that will depend on the intrinsic and extrinsic factors. For example, the nutrient content of the fish. So of course, specific organisms prefer specific nutrients. So if the nutrients they require are within that fish, or rather that product, they will be present. It will encourage them to grow. And then the extrinsic factors. How are you keeping that particular fish? Are you keeping it in a modified environment? Are you keeping it at a normal environment, which is aerobic? Are you keeping it at a higher temperature or at a lower temperature? All those will, both those factors will either select for or select against a particular organism. So for this study, we the particular environment, of course it's easier to alter the extrinsic factors. The intrinsic factors, there is very little you can do about them because that is part of the actual product. So the extrinsic factors, factor that we're interested in was aerobic storage. So and from literature, at aerobic storage conditions and at lower temperatures, it has been identified that Pseudomonas species and Shewanella species are the main organisms that spoil fish. So Pseudomonas will cause the generalized spoilage, while Shewanella will impart the bad odors and off flavors in the fish. So the objectives of this study was to monitor growth and then determine the lag phase, duration, maximum population density, as well as growth rate at different temperatures, and also to develop a secondary model for Atlantic salmon spoilage. So the methods, quite simple. We obtained our samples from a fish store in Hobart, and whereby their fish are harvested, then filleted after 4 days, and then at harvest, vacuum packed. So by the time we have the fish in our labs, they are about 5 days post-mortem, so which also represented our time zero. So when you see time zero, that means 5 days post-mortem. So it's at that time before we begin exposing them to the different temperatures that we wanted them to grow. So samples were transported on to the lab on ice, and of course kept in plastic containers with the wire racks, as you can see in the picture, and then stored at those temperatures. So for sampling, we got around 20 grams of the fish, which is approximately 2 by 3 centimeters width by length. And then to that, we added 180 grams of 0.85% saline peptone, which was also the first dilution. So we allowed 2 fillets for each temperature, okay, at least at the time. So at one particular experiment running, would use 2 fillets. So, and each fillet was sampled twice. So samples were homogenized in a stomacher and then later serially diluted to obtain a desired dilution. So for media and counts, like I mentioned, Shewanella and Pseudomonas are the ones that have been identified as specific spoilage organisms. So we're interested in Pseudomonas counts as well as Shewanella counts, and also total viable counts. So to obtain the total viable counts, we used standard plate count agar, which we'll select for everything that is growing at that moment. So initially we used pore plates as well as surface plates, but I thought that pore plates were better because they are easier to count, and you can actually have lots of numbers on a plate, and it's quite easy to count without being worried about colonies diffusing. So subsequently I used pore plates to determine my total viable count. So for Pseudomonas counts, we used CFC selective media, which is it's specific for Pseudomonas. It will only select Pseudomonas, and then we'll select against the rest. Then for Shewanella, Shewanella produces hydrogen sulfide, which of course has been identified as the one that causes a bad odor in the fish. So that particular hydrogen sulfide, the bacteria that produce that hydrogen sulfide produce black colonies on the iron agar. So this is some sort of differential media where it's easy for you to tell which one is which and which one is not. So plates were done in duplicates and incubated at 25 degrees centigrade for 48 hours. So for data analysis, of course we had the viable counts, which we converted to log CFU per gram, and then we fitted the data by using DM-Fit curve software to determine lag phase duration, growth rates, and maximum population density. Then to generate the secondary model, we used the growth rates and transformed them into square roots, and then plotted them against temperature, and then we fitted line. So our results, so these are the initial counts. As you can see, TVC, which were our total viable counts, these ones, and then Pseudomonas, there was hardly any difference, and statistically, oh, what's that? Sorry. There is this O here. It's supposed to be a sign. I think the computer changed it. Anyway, so as you can see, of course, it's quite obvious that the two counts were not much different from each other. But even then, statistically, it shows that there was not any difference between the two. However, Shewanella had up to about 4 logs. This one here had up to around 4 logs, different from the other two counts initially. That is looking at all the 10 samples that we had in the lab. But, pardon? Okay, so the results that I got, the lag phase is quite prominent at 0 degrees centigrade. We didn't observe any lag phase at 10 degrees for Pseudomonas and total viable counts. Then Shewanella had a lag phase of up to 10 degrees centigrade, rather at 19 hours at 10 degrees centigrade, and varied with samples at 6 degrees. Lag phase varied at 6 degrees because there was one sample that had unusually high counts. So we didn't determine the lag phase at that temperature. So maximum population density was the same for Pseudomonas and total viable counts. Then Shewanella had about 2 to 3 logs less than total viable counts and Pseudomonas. And then also days to maximum population density decreased with increase in temperature. So this is the summary of what I found, showing the lag phase, the maximum population density, where you can see total viable counts and Pseudomonas counts are quite similar, while Shewanella counts are quite less. So these are the representative curves that I generated. As you can see, the above two, which is TVC and Pseudomonas, they are quite the same, while this one is quite different from the other ones. You can see a longer lag phase and quite prominent, and then a less maximum population density than the above two. So growth rates, if you look at this point here, this, if you look at the growth rates here, this is Shewanella, this is Pseudomonas, and this is total viable counts. So generally, Shewanella had a higher growth rate, followed by Pseudomonas, then total viable counts at all temperatures. It's this point here that changed the trend at 6 degrees centigrade. So according to the linear regression fit, if you get the Pseudomonas counts, that means that you have the changes in Pseudomonas counts are attributed up to 90% temperature change. The same applies for TVC and Shewanella at those percentages indicated. So how does my research relate to that in literature? For Pseudomonas and total viable counts, all that I found, the initial counts, the growth rates, and the maximum population density, they were all similar to those that I found in different literature. While Shewanella, it had the highest growth rate. It also had the lowest initial counts, but the only difference with what I found in literature was the maximum population density was quite less, by 1 to 2 logs. Failure to reach that maximum population density may be explained by Pseudomonas antagonism, which does occur when Pseudomonas is growing with Shewanella. And also the plates that I used in my study were spread plates instead of pour plates. So these other studies which had different results with Shewanella used pour plates instead of spread plates like I used. So these are my conclusions. Lag phase duration increases with increase in temperature. Total viable counts and Pseudomonas counts, rather total viable counts and Pseudomonas have the same growth rate and achieve the same maximum population density. Shewanella has a faster growth rate, though it initially begins with the lowest counts, and it also achieves its maximum population density at an earlier time than total viable counts and Pseudomonas. And also, Shewanella minimum level of detection was at 2.8 logs. So for many of the samples, initially I could not get anything for Shewanella, though it doesn't mean that they weren't there. They were there but could not be detected. So 2.8 logs is the level at which you can detect them using yes and that. And those are some of the references.